Warm greetings. Welcome you all in CEC-UGC Gurukul live lectures. Learners, today in this lecture we will discuss about the chemistry of biopolymers, role of biopolymers in organisms and chemistry of nucleic acids, especially chemistry of DNA and RNA. So, before describing the chemistry of DNA and RNA, we must understand what are biopolymers. Learners, biopolymers are the polymers produced from the natural resources, and also biopolymers can be biosynthesized by the living organisms, and that biopolymers which can be produced by living organisms play a very important role in human biological system. Example, rubber. Rubber is a natural polymer because it is also made up of a monomeric unit, that is isoprene unit. But learner, rubber is not a biopolymer. Rubber is a natural polymer, but it is not a biopolymer. DNA, RNA, proteins, carbohydrates, collagen, all these are biopolymers because these biopolymers play a very important role in our biological, chemical reaction and biological processes. Biopolymers nowadays play a vast applications in industries, like they can be used in tissue engineering. Also, biopolymers nowadays are using for the formation of or the synthesis of nanoparticles. Example, collagen, which is a protein, and chitosan are used in tissue engineering. Also in many medical applications biopolymers are using. Biopolymers are the polymers that are produced from living organisms such as plants, animals and humans. So, it is very important to understand the biopolymers are used by the animals, plants and humans during many biochemical reactions, and these biopolymers produced by cells of living organisms, and all the biopolymers contain monomeric units, and that monomeric units covalently bonded together and to form a larger molecules. Learners, on the basis of their monomeric units, biopolymers can be characterized or can be classified into polypeptides. We know all the proteins are polypeptides, and that monomeric unit of that proteins is amino acids. Example, collagen, actin and fibrin, these are polypeptide. Similarly, another type of biopolymer is polysaccharides. These are the polymer of carbohydrates like starch, sugar, cellulose. And another very important biopolymer which is produced by human or our biological system is polynucleotides. Learners, these polynucleotides are long polymers which is composed of one or more nucleotide and nucleoside monomers. So, today in this part we will discuss about the chemistry of nucleic acid, especially what are RNA and what are DNA, what are the bases present in RNA and DNA, and how can we draw the structure of RNA and DNA, and which sugar is present in RNA and DNA. So, it is the classification of biopolymers on the basis of monomeric acid present in the biopolymers. Similarly, there is another way by which we can classify biopolymers on the basis of their origins. So, these are the classification on the basis of monomeric unit. Similarly, on the basis of their origin, that can be natural, that can be synthetic. So, these are the natural polymers, and synthetic biopolymers, that is polyallyl acetic acid, that is synthetic biopolymers. And why this is synthetic biopolymer? Because that contain a monomeric unit, which is a lactic acid, and that monomeric unit or lactic acid derived from the fermentation of corn, sugarcane and many agricultural waste, or we can say biomass. Similarly, on the basis of their origin, another important type of biopolymer is a microbial biopolymers, that is derived from the microorganisms. So, these are the some naturally derived polymer and their structures. These are the polynucleotides, these are the polyhydroxyalkanoids. Polyhydroxyalkanoids are microbial biopolymers. If we talk about the naturally occurring biopolymers, the biomass, especially wood, composed of three types of macromolecules. One is cellulose, hemicellulose and lignin. Cellulose and hemicellulose are polysaccharides, and lignins built from aromatic units. So, there are very many aromatic units joined together to form a lignin molecules. Polyhydroxyalkanoids, PHAs, are polyester, which is a synthetic biopolymer produced in nature by microorganisms. And learners, these PHAs are emerging class of bioplastics produced by bacterial fermentation using bio-derived feedstocks. So, the microbial fermentation of sugars, glucose and vegetables, we can obtain polyhydroxyalkanoids. Though these biopolymers are having very important characteristics, they are biodegradable, biocompatible, renewable, they are non-toxic. So, due to all these properties, they have a versatile applications. So, they used in medicinal industry, they used in food industry. So, many industry, they are having a many industrial applications. Now, what is the role of biopolymers in organisms? So, these biopolymers can act catalyst or can catalyze the reactions which can provide the energy or other nutrients to the organisms. Another important role is they can protect organisms against the attack of other cells, storage of carbon, and also they can negotiate of adhesion to surface of other organisms. These biopolymers can articulate with the environment and other organisms, preservance and expression of genetic informations. So, the biopolymers formed during the life cycles of animals, bacteria, fungi and green plants, and these biopolymers are DNA is a biopolymer of nucleotide. And learners, DNA is a very important biopolymer, and this DNA is main constituent of genes. So the biopolymers generated by living organisms are biodegradable polymers. DNA and RNA, deoxyribose nucleic acid and ribose nucleic acid, both are self-assembled anionic macromolecules. So the nucleic acid based biopolymers have significant potential as functional material for drug delivery, biosensors and scaffold for many biodegradable materials. They are chemically modified nucleic acids biopolymers which is used in biosensing, gene therapy or gene engineering, also controlled drug release biomolecule detections. So these are the very important points or role of biopolymers in organisms. Example I would like to explain here: seaweed. Seaweed is a type of plants or algae, species of plants and algae which is grow in sea, river, lakes or other water bodies. So the seaweed is a sustainable source for bioplastics and it is also very emerging class of biopolymers. So using seaweed, now industries are obtaining bioplastic which is the sustainable bioplastic, because the seaweeds are high biomass rich in polysaccharides. These are agroses, alvin and fucoidal green and brown algae. So these are the species of seaweed which are having a diverse applications as a biopolymers. So this is the structure of seaweed you can see on your screen, and these seaweeds which are the plant or algae species which is sustainable source for bioplastics. Learners, the bioplastics we can obtain from the seaweed is a edible plastic also sustainable plastic, edible plastic, dissolved in boiling water, used as a sachet or pouch, edible cups, plastic bags, wrappers which is made up of cellulose, packing made from seaweed are biodegradable and sustainable, and these are used as a thickening of gelling agent in cooking. So this is the reference, you can go through the reference and can know more. Now structure of polymers of nucleic acids. What are the structure of nucleic acids? Learners, nucleic acids are nucleotides, polynucleotides, we can say polynucleotides, and nucleic acids are genetic material of all living organisms. The monomeric units of nucleic acid is deoxyribose nucleic acid, that is DNA, and DNA is the carriers of all the genetic information in all living organisms. And other monomeric unit, that is the ribonucleic acid, RNAs, it is an agent and responsible for the transferring of all genetic information for the protein synthesis. Learners, this nucleic acid also participate in synthesis of coenzymes. Coenzyme A, nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphates and flavin adenine dinucleotides. So all these are the coenzymes, and these are the enzymes which are used to generate energy and also used for the repairing and formation of bodies. Learners, this is the structure of on your screen, you can see flavin adenine dinucleotide. It is a redox active coenzyme, and this is a very important coenzyme. This enzyme is associated with protein and involved in enzymatic reaction in metabolisms. Learners, this FAD, flavin adenine dinucleotide, exists in 4 redox states. One is flavin, N5 oxide, quinone, semiquinone and hydroquinone. So, the flavins are ubiquitous class of redox active coenzymes. In human, the flavins are obtained as a vitamin B12 from our diet. So, this is the structure of coenzyme A. Coenzyme A is a very important enzyme which is used to generate energy through the biochemical process, and this coenzyme A conversion of fats, proteins and carbohydrate into metabolic energy. So, this coenzyme A, you can see the structure, it is contain of 5 parts. One part is sugar plus base, other part is we can see thiol group, acetyl group. So, this coenzyme A is a very important enzyme and it is used to generate the metabolic energy. Similarly, NAD plus, which convert food to energy and also may help in maintaining the DNA integrated. So, on your screen you are looking the structure of coenzyme A and nicotinamide adenine dinucleotides. Now, these are the some important points you have to learn, memorize always. In most of organisms, the genetic information, all the genetic information is stored in DNA, that is deoxyribonucleic acids, and these information transcripted into RNA, and from that RNA the information can then translated for the synthesis of all the proteins which is needed for the cellular structure and function. So, this is the very important point. The DNA stored all the genetic information and that genetic information is transcribed into RNA, and here the information can be then translated for the protein synthesis. Now, nucleotides, nucleosides, DNA, RNA, all these are the biomolecules because all these are the essential for numerous biological functions in humans. Also act as a energy storage, maintaining and transferring genetic informations. Also regulation of various aspects of metabolism, act as signaling, used as an antioxidant, and deficiencies in their synthesis led to death. Also, overproduction or decreased elimination of nucleic acid derivatives lead to directly medical conditions. Now, what are nucleosides? We have talked about here nucleotides, nucleosides, nucleic acids, RNA and DNA. So, learners, nucleic acids are having nucleotides, nucleosides, DNA and RNA. Now, we will discuss what is nucleoside. Nucleoside, these are the two different terms. One is nucleoside, another is nucleotide. So, what is the nucleoside? Nucleoside is the structural subunit of nucleic acid and it is obtained by chemical or enzymatic decomposition of nucleic acid, and the nucleoside is composed of two components. And what are these two components? Base plus sugar. So, there is a two components, one is base, other is sugar. So, sugar is also here pentose sugar, right, and sugar can be ribose sugar or deoxyribose sugar. So, on the basis of sugars, if there is a deoxyrized sugar, we call this deoxyribose nucleic acid. If the sugar is ribose, we call this RNA ribose nucleic acid. So, the nucleoside having bases and sugar. Now, what are the bases present in the nucleoside? So, there are two types of bases present in the nucleoside. One is purine base and another is pyrimidine base. So, on your screen you can see the basic structure of purine base and pyrimidine base. So, as in purine base you are looking here, there is a two ring, it is a cyclic structure, it is a aromatic structure, also heterocyclic structure. So in purine there is a 2 ring, one is 6-membered ring which contain 2 nitrogen atom, and another is a 5-membered ring which is also contain 2 nitrogen atom. Similarly, it is a basic structure of pyrimidine base. So in pyrimidine base, this is also 6-membered ring and having 2 nitrogen atom in their molecules. So here always remember 2 types of bases present in the nucleosides and nucleotides, purine bases and pyrimidine bases. Learners, if we talk about the numbering of purine and pyrimidine bases, the carbon and nitrogen atom of purine and pyrimidine are numbered anti-clockwise. So it is always remember, you have to write numbered the nitrogen and carbon anti-clockwise. Pyrimidine ring numbered are anti-clockwise, but basically normal nomenclature, the pyrimidine ring having a clockwise numbers, and also both ring are planar, and this characteristics of ring help in their parallel stacking, strengthened by hydrophobic interaction, which help the DNA strand in attaining helical structure. This is the structure of purine base. So it is adenine and commonly it is represented by A. So purine base is a 9-membered double ring structure with 5 carbon atom and 4 nitrogen atom. So it is a 6-amino purine which is adenine A. Another is purine base is a guanine. So guanine is a 2-amino oxopurine. So all these are having, all the purines having one 6-membered ring and other is a 5-membered ring, and it is a imidazole ring. Another type of basis, that is a pyrimidine base. So pyrimidine base, this is the structure of, on your screen you can see structure of cytosine. It is represented by capital C. So pyrimidine base is also 6-membered ring having 2 nitrogen atom, 4 carbon atom. It is structure of cytosine, it represented by capital C. Similarly, it is a structure of thymine and uracil. So these are the pyrimidine base. Learners, on the basis of we can differentiate in the DNA and RNA. So DNA having thymine base, and in RNA thymine is replaced by uracil. So there are 2 different bases present in DNA and RNA. This is the structure of pyrimidine base. So all the bases present in nucleic acid are represented by first letter of their name. So it is the structure of uracil, adenine, guanine, thymine and cytosine. So the pyrimidine bases in nucleotide or nucleic acid present are CTU, cytosine, thymine and uracil. Similarly, the purine bases present in nucleic acids are adenine and guanine, and all the 5 bases present in abundance in nucleic acids called major bases, and these are the major energy carriers, precursor for synthesis of nucleotide cofactor NAD. So learners, in this part we had discussed about the biomolecules, what are biopolymers and what are the difference between the natural polymers and biopolymers. As I have told you, the biopolymers are the polymers which are biosynthesized by the through the biochemical reaction by living organisms. Thank you.